today. Today, my plan is to talk about my work leading up to that and introduce you to the CDA project that I'll be working on for the next 5 years. My past, just like Dakshin nicely outlined for me, my bachelor's degree through my dissertation was all conducted at the University of New Hampshire, where I studied contributions of the thalamus to what was believed to be prefrontal dependent executive functions. I manipulated and recorded thalamocortical pathways in rodents performing various executive function tasks. Then I came to UCSD to work with Dr. Dakshin Ramanathan, and the goal was to learn large-scale recording techniques to study communication in preclinical rodent models. My time at the University of New Hampshire was also a teaching intensive program. So along with balancing research, we were the instructors of record for psychology and neuroscience-based courses. And then in my final year of that program, I received a dissertation year fellowship to buy me out of teaching responsibilities so I could focus on research. From the collective experiences at the University of New Hampshire and those I've had at UCSD, my research goals is kind of summarized below as that my research aims to elucidate how neurons communicate information underlying cognitive processes across networks and examine how communication is disrupted in the case of neuropsychiatric disorders or brain injury. So the transition from University of New Hampshire to UCSD meant a few things. One, cold weather. It's a beautiful San Diego weather. My dog and husband are both very aligned on which one they prefer. So that's good news for all of us. More importantly, though, it also meant transitions in the way that I was tackling my research design and the hypotheses that I was creating. Namely, that meant that my perspectives changed a bit from a single neuron view to encompass larger scale neural networks. This also went hand in hand with designing research that changed from a basic science view to a more translational perspective. And I think being positioned in the psychiatry department specifically helped to shape some of those ideas. Therefore, the outline of my research that I'll talk about today encompasses these transitions, and in my scientific approach, spanning my doctoral and postdoctoral research in the following areas. First, I look at how brain network communication normally occurs. Second, I can frame this in how communication is disrupted in the case of traumatic brain injury, which I'll really highlight today, or different psychiatric disorders. And then third, I can think about how we can use that information about brain communication to remediate some of those deficits. My dissertation used single unit electrophysiology to characterize thalamocortical circuits and causally manipulate thalamic nuclei input to PFC. This describes well how I study discrete brain areas and their limited connections. And then as a postdoc with Dakshin, I've applied large-scale electrophysiology techniques that are mostly widespread local field potential recordings to study markers of impulsivity across different brain networks. So more of a distributed view of neural networks. My primary measure of interest in these experiments, and what I'll mostly be focusing on today, is electrocellular electrophysiology that happens in vivo while rats are awake and behaving and performing different operant tasks. This is just an example of one of those such tasks. I'll describe a few different ones today, where rats are either performing nose pokes or lever presses in an operant scenario. We also have done some spontaneous behaviors, although I won't mention those today. And then as the rat's performing, I'm recording brain activity either at the level of single units or at the level of local field potentials. And we're time locking that activity as shown in C to different task-related events. And we have one of our male models showing a single unit implant. These recordings can span a very chronic behavioral time frame if needed. We have actually extended some studies beyond even 12 months. I only bring this up because it is advantageous, especially when we're talking about neuropsychiatric disorders or brain impairments from TBI, where patients often are suffering long-term consequences. And so this technique that we've developed is great for looking at that time frame. My dissertation research was supervised by Dr. Robert Mayer, whose prior research began to debunk the thought that the thalamus was simply a passive relay passing on sensory information to cerebral cortex. He investigated this problem first in human postmortem tissue, where he was finding that thalamic damage was associated with amnesia in humans. He was mostly looking at thalamic damage that was caused from thiamine depletion in Korsakoff's patients, and then started to translate this work into rodent models as well. Simultaneously, others were investigating this, and anatomists were revealing that there are actually dense reciprocal connections between prefrontal cortex and thalamus, which would argue against a simple feed-forward mechanism. And now, of course, we recognize that there are two types of thalamic nuclei. There are first-order thalamic nuclei, which are the driving input from sensory areas and does act as a relay in this feed-forward mechanism. But there's also these higher-order nuclei, which receive driving inputs from cortex and do have a modulatory role on cortical information. Most of these make up the central nuclei of the thalamus, shown here on a coronal brain section of a rat brain. And I'll talk about those more specifically as my dissertation focused on them. Dr. Mayer and others like Yogita Chudasama and Ann Mitchell in primates were showing that lesions to central thalamic nuclei had damaging effects on many executive function behaviors, one such being working memory. To move beyond this lesion work, we began using electrophysiology as a way to better classify the role of higher order thalamic nuclei in respect to a prefrontal cortex. Our primary task that we used to explore this was a delayed non-match to position task in the rodents that they would learn to perform in an octagonal arena. And I have a little video and description of that I can play in just a second. We picked this task because it had the working memory feature, which was one of the highlights that we knew would be thalamic dependent based on the lesion literature. But we also liked that it had a lot of other components. We made it dynamic such that the start lever was randomly selected from the 4 locations at each trial, and the sample lever was then randomly selected from a left or a right turn. So it was dynamic. The rats couldn't just memorize a fixed pattern, and that led to some interesting behaviors that we could analyze from this. So I'll go ahead and try to play this short video. There should be audio in a second. If you don't hear it, let me know, and I will gladly speak over it.
This video shows 3 trials of the delayed non-matched position task. In one session, rats must complete 61 trials or time out after 60 minutes. The trial begins with a start lever extending from one of 4 possible lever locations. Upon pressing, a sample lever extends to either the left or right of the start location. The rat is reinforced with water and must return to the start location for a short delay held at 3 seconds. Both levers to the left or right extend. A correct choice is the non-match to sample location where water reinforcement is delivered, and a new trial begins. The start lever is randomly selected between trials such that the rat cannot memorize a fixed pattern. The light indicates when water is delivered. If the rat chooses the error location, he is given no reinforcement and must correctly choose the opposite lever before a new trial can begin.
Great. So that's just, you know, an example. It's always kind of fun to show the rat's behavior of what it looks like while we're recording them in the task. The yellow little action potentials that just popped up signify what this actual example neuron that was recorded during this day of behavior was doing during this task. And this particular example was what we would go on to deem a movement cell, where it was firing between all patterns of movement in the task. So from start to sample, from sample to delay, delay to choice, etc. So that just shows one example of how we're able to time lock the firing rate of the task and elucidate the properties of the neurons. We started by recording from areas of the medial prefrontal cortex in rats during the DNMTP task. We ended up sampling over thousands of neurons and were able to classify 12 distinct response types based on the temporal properties of how the neurons were firing during this task, or what windows of time they were firing in this task. These distinct response types were associated with actions, such as the movement cell that I just described, or lever press actions in the task. And also some are related to outcomes, either anticipating a reinforcing event, signaling a reinforcement outcome, or signaling an error event. What I'm showing you here is just a synopsis of the population activity. So all of these thousands of neurons, their average Z-score activity. The main point here is that they all are specialized to fire in a discrete window of time, some overlapping, but all distinct. Other researchers recording from prefrontal cortex around this time, before this time, since this time, have found similar responses. So this wasn't necessarily surprising, but the variety that we see in one task maybe wasn't captured yet in these other tasks. And so that was just one reason that we did like the DNMTP task as well. The second main takeaway from this study was that the prefrontal neurons we collected also had spatial mapping features. The firing rates of the neurons were not surprisingly tied to the location of events, but some neurons were actually spatially specific, only firing at one distinct location. This example, let me get the laser pointer going. A is showing you a lever press cell. Not surprisingly, the red indicates the highest level of firing rate for this cell in the octagonal chamber. For the lever press response, that's happening at all 4 of the lever locations. The surprising finding we made was that some are spatially restricted. This is an example of a neuron that is another lever press cell, but actually only cares about one lever location. And then the third finding that we highlighted throughout these studies was that we did find some evidence of subregion specificity. Generally, more dorsal regions of prefrontal cortex were action related and ventral regions were more outcome related. And that's since been replicated in future studies as well. Next, when we had this basis of what PFC was doing in this task, what we were really excited about was to go ahead and investigate central thalamus. So in separate animals, we did a similar design where we recorded behavior in rats performing the DNMTP task, while we recorded thalamic activity, mostly from the mediodorsal nucleus and intralaminar nuclei, both central thalamus areas. First, we found similar temporal responses as were observed in the prefrontal cortex. Thalamus had mostly movement-related and reinforcement-related outcomes, and not quite as much diversity as prefrontal cortex had, but still the response types mapped on in a really temporally specific manner that was consistent with what we were seeing in prefrontal cortex. Second, we also were able to observe this phenomenon of spatial mapping that existed in thalamus, and it actually existed in somewhat a greater extent than PFC, which was a little surprising. Some cells were very specific and only tracked movement, for instance, towards the reinforcement lever location. So it seemed like thalamus might even be better situated for integrating spatial information with reward-related locations. And then thirdly, there was also some evidence of subregion specificity that existed in the thalamus. Intralaminar nuclei was more delay related and more action related, and mediodorsal nucleus was more reward related. This actually shows a nice correlation between what the anatomy shows between these subregions and their connections to prefrontal cortex. So medial dorsal innervates more heavily at the ventral areas of PFC, and interlaminar more heavily with the more dorsal regions. And so we were able to see that with the cell type distribution as well. It thus seems reasonable to hypothesize that central thalamus is specialized to monitor signals from prefrontal cortex related to autonomic and limbic function, as well as motor function. And consistent with this view, the thalamus may have a role in regulating reinforcement guided responding, previously thought to be only prefrontal dependent. Now that we had support of this hypothesis, the next question was, how is central thalamus modulating cortical activity? Is it controlling it or shaping it in any way? One way that we have investigated the influence of the mediodorsal thalamus on prefrontal cortex was to perform an inactivation study by recording cells in prefrontal cortex while injecting mucimol into the central thalamus to see how inhibiting the cellular response in thalamus would affect the cortical response types that we were able to record. This was a unilateral manipulation, and thus we were not expecting any global behavioral deficits in the DNMTP task. That has been shown with lesion studies that the unilateral thalamic lesions didn't affect behavior. So we weren't really after capturing behavioral changes, really specific to what the cell types would be doing. So cells in prefrontal cortex were recorded while we were injecting mucimol, which is a GABA-A agonist, into the medial dorsal nuclei of thalamus to see how inhibiting cellular activity in MD would shape the neuronal activity in cortex. Here I'm showing you an example of one particular cell that went through this paradigm that we recorded. Day one, we always had a normal recording day where we just captured the cellular activity without any influence on thalamus. You can see the different properties of the cell and how we classified it as a cell. And then this is its event-related activity relative to the start lever press in the task. So this was a lever press response where the neural activity is heightened around the time of the lever press. On day two, when we inject the mucimol to inhibit thalamus, the cortical response type drops out. So we're holding the same cell across days, and we verified that in a number of ways based on its cell type, cell shape, and where it was located. And so the event-related activity drops out and firing rate is weakened when we inactivate thalamus. And then we try to always have a recovery day where we're still holding that same neuron and seeing, again, without doing anything to thalamus, does that activity respond like it did in day one? Does it come back if it was knocked out? So this is one example showing a huge knockout. When we remove thalamic input, this cortical cell type does not have any event-related properties and loses a lot of its firing rate. This was not always the trend. Sometimes we would see an amplification on day two. Sometimes we'd see no change. But the overall pattern of change out of all the neurons we collected was a significant decrease in the thalamic response on day two. So this study from our lab and many others in the field, including Kellendonk's lab, who's done some really nice chemogenetic dissections of the circuit, told us that MD does influence cortical activity. I was specifically able to replicate this effect using both unilateral lesions and recording the cell types, and using dreads to innervate the circuit while recording the cell types in medial prefrontal cortex. And we were showing similar effects. So yes, I did a lot of work to explore this circuit of thalamic input to prefrontal cortex. And yes, the thalamus does seem to have a role, yet complex functions are not generated by one or even two discrete brain areas. Thalamocortical operations are actually part of a much bigger network that controls cognitive behaviors. Therefore, I was wondering how I could put this into perspective to better characterize neural network problems, especially in the case of neuropsychiatric disorders or brain injury. In order to do this, I thought I would need to zoom out and investigate cortico-thalamic striatal circuitry, which are largely involved in reward-guided behaviors and are dysfunctional in a number of neuropsychiatric disorders. To study network-wide changes, I would need to supplement the single unit studies with a more macroscopic, zoomed out level of investigation, which also holds translational potential, perhaps to humans. And so local field potentials are one level of investigation, which might be favorable for this type of investigation. So now I'll move on to talk about communication in distributed brain areas. With Dakshin, when I got to UCSD with Dakshin and the team at NEAT Labs there, we developed a new way to record brain activity that branches multiple networks. So we developed a way to record 32 different brain areas simultaneously in the rat that spanned networks like visual networks, motor networks, reward networks, memory networks. And as you can imagine, there's many benefits to this method. I'll list just a few. One is the translational potential to human EEG. We actually have papers as a lab identifying similar markers in the rodents using this method, and identifying similar markers in humans with EEG. So LFP holds that translational potential. Second, it eliminates the need for strong a priori hypotheses because your areas of investigation really aren't limited to one or even two brain regions. Therefore, sometimes even if I think something like visual cortex might not be an area of interest or be responding in this task, I'm actually not limited. I can go back and post hoc explore it. So we're much more likely to find things that might have been missed before. Lastly, LFP bridges microscopic and macroscopic layers of investigation and sits at a nice intermediate to investigate some of these network-wide changes in the brain. Our goal to use this approach was first to identify physiological biomarkers of cognitive behaviors, and second, to investigate how these biomarkers are changed in the case of neuropsychiatric disorders and traumatic brain injury. Our thinking is that if we can identify a robust and reliable biomarker, we would have the potential to predict or track treatment outcomes even. Just a really brief introduction to local field potentials for anyone who might not be as familiar with them in this talk. Basically, neural activity is not random. It oscillates at periodic frequencies that coordinates communication within and between distributed brain regions. There's several oscillatory bands that span across frequencies. There's delta all the way up to very high frequency gamma activity. And these frequencies can influence each other by modulating their phase and or their amplitude. Phase synchrony is a reflection of how coupled two brain areas might be. Here I'm showing just an example of two hypothetical brain regions, and their waves are very synchronized. So that might be an instance where these two brain regions are interacting and sharing information versus these two that are out of sync. And then C is an example again that I showed previously of how we can actually time lock the local field potential activity to task-related events. We can time lock the data to a rodent's response in a task, to reward delivery, to trial onset, for example. And we can determine what the brain activity was doing during that discrete window of interest. In this figure, gray is the cumulative LFP trace, and blue is just highlighting the filtered out beta frequency. Each of these frequency bands are predicted to coordinate different aspects of behavior through long-range coupling in different functional networks. Changes in the oscillatory activity at these discrete frequency bands can indicate disease states like Parkinson's disorder, depression, autism, attention disorders, and schizophrenia. We've used this approach now to identify physiological biomarkers associated with different forms of impulsivity. And impulsivity was of interest to us because it's known to be a central feature in many neuropsychiatric disorders like schizophrenia, attention disorders, substance abuse, and even in traumatic brain injury. We picked 3 tasks to start to focus on, and we picked them because they all sit on kind of this scale of impulsivity, from being more motor impulsivity related to more reward impulsivity related. Ironically, today I'm actually not going to highlight the differences of these tasks. I'm more going to argue for one central feature that they all have in common, which has to do with reward outcome and the presence of reward in each of these tasks. We used each of them to our advantage to tell us something a little bit different about reward guided behavior and how the rodents might be using this reward information to shape decision making on each task. So in the rats, we've used our brain-wide 32-channel recording technique to capture oscillatory activity, signaling reward-guided behavior in these 3 different tasks. The spoiler alert that I will delve into more detail is that we find beta frequency activity during the reward outcome period reliably in all 3 of these tasks. And so we're building this hypothesis that beta frequency activity specifically might be monitoring the reward outcome and using that information to help shape future decision making. These tasks also capture various aspects of motivation, reward learning, cognition, attention, each of which deserve more time than I have to discuss today. And really, I will just be focusing on these tasks for the purpose of a reward outcome type of discussion. So just to describe each of these very briefly, in the go wait task, which is more a motor impulsivity task that we used, the rats either have to perform or withhold from a nose poke depending on cue presentation. So if rats are shown a go cue, they simply have to respond in one of the nose ports within 2 seconds to receive a reward. If the rats are shown a wait cue, they simply have to withhold from responding for 2 seconds and then can respond to receive a reward. If they fail to withhold a response, they will not get reward. So this sets up a nice comparison of rewarded or not rewarded trials. It's of course way more dynamic than that, but for the purpose of this investigation, it's just a comparison of presence or absence of reward. Next, we also studied a probabilistic reversal learning task in which rats have to discriminate between high and low probability nose ports and flexibly respond when those nose port contingencies are updated. Rats are presented a choice between 2 response ports. One is a high probability response. It will lead to water 80% of the time. The other will only lead to water 20% of the time. Once rats choose this nose port in 8 out of the last 10 responses, then the contingencies will reverse and we can count how quick rats are to catch on to that reversal. The last task that we use is a temporal discounting task that tests how long the rodent is willing to wait for a large reward opposed to a small reward that's delivered immediately. The large reward, which is a 3x times reward, may be delivered immediately, or it might be delivered at a variable delay up to 20 seconds. The low value reward is always delivered at an immediate, or is always delivered immediately with no delay. The delays in this task were changed between sessions, and there were no within session variances. Okay, so what do we see for distributed brain activity when we're recording from this task? First, in the go wait task, we found that beta and high gamma frequencies signal positive valence, so when reward is delivered. And we started this investigation at the lateral orbital frontal cortex electrode, which is a cardinal reward processing region. We measured the power during the reward outcome period of 3 different trial types, correct trial types on both go and wait cue, and incorrect on the wait cue. There were so few go Q incorrect trials that we had to leave them out of this investigation. So on either the GO or the weight Q correct trials, we see elevated beta and elevated high gamma activity, and that drops out when there's no reward present on the incorrect trials. This trace of normalized power also shows this difference just in a different way, plotted over time, but the peak for the correct trials when there's presence of reward is higher and longer in duration than the weight incorrect Q trial. Not only is this pattern seen in the lateral orbital frontal cortex, but it's also widely observed throughout other cortico-striatal brain areas. We do get a main effect of electrode, but that's driven here by the basolateral amygdala electrode, in which the weight incorrect trials also have a heightened beta activity compared to the other electrodes. So this is the first evidence of at least two frequency bands that might be coding reward-related information in this task. And we wanted to expand that to see if that held true across these other tasks of reward-guided behavior. So next, on a probabilistic reversal learning task, beta activity was also heightened for reward delivery, and importantly, was strongest for expected reward. On the high probability choice, beta activity was stronger than rewarded trials even on the low probability choice, showing that not only does beta frequency maybe modulate with reward presence, but also the expectancy of reward elicits a different response at the beta frequency. Again, this was true not only at the lateral orbital frontal cortex electrode, but it's seen in other regions like the anterior insula, lateral orbital frontal cortex, which is the same above here, nucleus accumbens regions, and ventral orbital frontal cortex. The main effect of electrode here was driven by the increased power at anterior insula and lateral orbital frontal cortex compared to other regions. So where the effect might be the strongest. Lastly, in a temporal discounting task, beta activity was strongest for the high value reward, more reward magnitude, and that scaled with subjective value. Subjective value on this task decreased over longer temporal delays. So as rats were asked to wait longer for the high value reward, not only did their behavior change where their preference for the high value reward diminishes, but that's also actually reflected in the power of this beta frequency activity. So we start at an equal match delay where the high reward beta power is much greater, and that slowly diminishes as the delay gets longer. Again, this trend is seen at the lateral orbital frontal cortex electrode, but is actually also consistent across many other areas of the cortico-striatal network. This is looking specifically at the difference between the high minus low value choices at each of these delays. So we see that nice steep decrease across many of these cortico-striatal regions. There's actually no significant effect of electrode in this data set. Therefore, we've accumulated evidence across 3 distinct tasks that indicate that beta frequencies may represent a robust, reliable indicator of subjective reward value that occurs in the cortico-striatal network. This is by no means only interesting finding from these tasks, and each deserve a much more thorough discussion. Some of the other frequency bands also showed interesting things at different task-related events, so happy to discuss further if anyone has any interest in that. But what I do want to highlight here is that the beta frequency activity we're seeing represents a great example of a potential biomarker that could be used as either a treatment option. We could try to remediate the circuits by heightening this beta activity, or it could be used as an indicator to track network disruption or predict treatment outcomes. And so to follow up on that, we can next, once we find a kind of potential biomarker like this, we can next ask ourselves how this biomarker might be disrupted by the traumatic brain injury, or in the case of neuropsychiatric disorders. As I mentioned previously, my awarded CDA is to study these effects in traumatic brain injury. And so that's just what I wanted to highlight today for the last piece of my talk. Okay, so for my CDA, I will be harnessing, again, this 32 electrode simultaneous recording in rodents. They'll be given severe, mild, or sham TBIs. We're going to use the controlled cortical impact model that I'll highlight in the next slide. And we'll investigate how TBI affects the large-scale neural recordings. So we'll do TBI, I will implant the recording electrodes. They'll go through behavioral training. In this case, I'm starting with investigating the probabilistic reversal learning task, where we know TBI has behavioral deficits. We'll do some chronic LFP recordings and end the experiment. Then I have actually also proposed to send the ex vivo brains through an MRI scan so I can collect diffusion tensor imaging. This will complement our functional network measures with structural measures of white matter integrity, and investigate those neural connections from a structural standpoint as well. And I'll also be able to perform histopathology on those brains after they've been scanned. My main hypothesis is that if prefrontal cortex communicates information about reward with its distributed striatal network, then damage to frontal cortex resulting from TBI will produce network-wide deficits in brain communication. So as I mentioned, the first surgical procedure will be a bilateral frontal controlled cortical impact injury. Controlled cortical impact is created by driving an impactor into the brain and is designed to closely mimic cortical tissue loss, acute subdermal hematoma, concussion, or even coma that occurs in humans. Its severity is influenced by the size of the impactor, speed, and duration of impact. I chose this model to start with because it's very experimental. It's very controlled by the experimenter. When we are considering pairing a frontal TBI with our large-scale recording apparatuses, I also do need to have an intact skull to attach the electrodes to, and so that was one of the requirements. Our future endeavors will be to test this in other models and some that might be more translationally relevant to our veterans, but we'll start with both mild and severe injuries here. And I will say, I should recognize that we are replicating injuries here known to produce probabilistic reversal learning deficits. We're working with our collaborator Cole Vonderhaar out of Ohio State University, who previously was with Catherine with Stanley, and both of them have shown that this particular injury leads to reward-guided deficits on various tasks, one being PRL. Okay, rats are going to be given a week to recover, and then the second surgical procedure will be to implant the 32 channel local field potential probes. The only thing I need to highlight here is that, yes, some of our electrodes are directly in the site of the TBI, directly in the area of the impacted tissue. Others are adjacent to, or actually some are very, very downstream in striatal areas. So we're able to study, again, a wide variety of electrode sites, some very focal and some very distal to where the lesion is being provided. Okay. So far, for pilot data, I have replicated the severe injury in approximately 20 male rats. We will need to repeat this in female rats, in the mild injury rats. There's still a lot to do, but we have the data for 20 male rats. We've performed the histopathological analyses. This is just the Nissl stain. And we've revealed, significant lesion volume loss, increased gliosis, lack of cell bodies that other investigators and our collaborators have previously found. We have also recorded then their behavior on the probabilistic reversal learning task. Performance is affected by the traumatic brain injury. The TBI animals are shown in red, the red squares. The TBI significantly reduced the number of reversals compared to sham. Both learn across time or improve across time. This is 11 weeks post-injury, but the TBI rats do that at a slower rate and never are able to quite perform the number of reversals that sham rats are able to perform. We did compare that against just the number of trials that they're able to perform, and there's actually no significant difference there, no interaction there. The other major deficit that we see are that TBI rats have a harder time discriminating. They take more trials to complete their first reversal than sham rats do. There was actually no reversals for the TBI rats occurring at week 3, but if we look at 4 weeks out, this is a very early effect that does seem to mediate across time, but TBI rats always take a little bit longer to get that first discrimination block than the sham rats. So here we're just replicating, results that many others have found. This is consistent with what a CCI TBI and frontal cortex kind of looks like with behavioral impairments. What I'm specifically interested in for the CBA is actually what is driving these behavioral deficits that we see on the PRL task. And I'll be examining that, primarily looking at the local field potential activity. So first, using again the lateral orbital frontal cortex electrode as a starting point, we see increased beta and delta frequency activity during the reward trial outcome. That's different in the TBI and the sham rats. On the no rewarded trials, we see no difference in behavior, just not much power is elicited across any frequency band. To put this into better perspective, we can run this as a contrast of the rewarded trials minus the no rewarded trials to look at the difference between trial types, or the difference when reward is present or not present. And this really highlights the effect at beta frequencies, where sham rats show a big difference between trial types, and that's diminished after a frontal TBI. So this lack of differentiation between trial types might be better shown with a shaded error trace. This is the same thing on the lateral orbital frontal cortex, just plotting the effect across time. Blue indicates when reward is delivered. So during this reward outcome period, sham rats show an increase in beta during the reward outcome. That is not there for no rewarded trials. So a clear differentiation in activity and power in the sham rats. What happens in the TBI is that gap between trial types is closed. So beta frequency is not differentiating at the level that sham rats are between the trial types. And this could be, one of the contributing factors that's causing a lack of reversals that we see behaviorally. Lastly, this effect is not unique to the lateral orbital frontal cortex. It's also observed in areas that are local, highlighted in red, local prefrontal areas to the lesion, areas that are more local, and areas that are actually very distal. Some striatal regions also show this impact. Lateral orbital frontal cortex and anterior insula seem to show the largest effects, mostly driven by the large increase in power in the sham rats for rewarded trials like we saw previously. I was going to be surprised if I had time to tell you about this, and I won't. I'll pause for questions instead, but I will just lay this out as a way to say that is how I'm investigating it in traumatic brain injury, but we can also do this same type of investigation for neuropsychiatric disorders or investigating potential treatment outcomes. So This is an example of a drug study that we're running, looking at methylphenidate and citalopram and their effects on impulsivity behaviorally, but then also what are they doing to the brain-based markers that we would track at this time. So I won't have time to go into those results, but simply another point to say how we might be looking at communication when it's disrupted in these cases. And then finally, my last point of interest as a researcher is more of a future goal for my research, and hopefully I can report back in a little bit to tell you more of these exciting findings. But the third point is how can we remediate brain communication once it is disturbed? And so this could be with pharmacologic treatment, as I showed kind of briefly on the previous slide. It could be with behavioral manipulations. We're currently doing this with our TBI collaborator, Cole Vonderhaar, in TBI animals. Or it could be through neuromodulation. The last aim of my CDA actually proposes to use a TMS-like approach in the TBI rats and measure behavioral and physiological changes after TMS to see if we can induce long-term changes in the reward network. So hopefully by the end of my CDA, I'll have some follow-up points to talk about with TMS-like approaches in the rodents. I just want to thank everyone for their attention today. I'll welcome any questions at this time, and for my collaborators at UCSD, all the members of NEAT Labs, previous and current postdocs, and my mentors at the University of New Hampshire. Thank you, and hopefully people have some questions.
Thanks so much, Miranda. That's great. I will see if there's any questions coming in, either in the chat. One thing I'll say is, given how many strong behavioralists there are in our department, including people on this chat, do you see, or what are your potential collaborative interests, or how do you see the things you're doing could be useful for people who are interested in impulsivity in the context of addiction or cognitive disorders?
Yeah, I mean, I think the thing that we are kind of most excited about at this time is just the potential for how our brain recording techniques can be used in a variety of these tasks. So I think proving that we can find a robust biomarker across 3 separate tasks that tracks is hopefully something that people in the department are excited about, and I think highlights the ways that I'd be interested in collaborating if it's a task of interest that you have, or if it's a disorder of interest that you have. This is a way that we can apply, brain recording techniques to study that. And then, of course, I'm excited to move into the traumatic brain injury field, where I think there's just much more work that needs to be done to classify different behaviors. The PRL was almost a safe choice, but I'm also excited to explore different paradigms and different models of that might not be quite as safe or guaranteed to find these robust behavioral effects.
Great. Since, let me see, there's 2 chats. Great presentation. I don't see any of the human TBI folks on here, and I know that's kind of a newer area for you. But what is your take on how we can apply results from rodent TBI work to clinical human TBI? I mean, what are the limitations and what are the advantages of using rodents, and kind of just your take on that?
Yeah, I mean, obviously, I'm highlighting a very invasive measure with the implants. And so that's kind of an obvious advantage to using our model. But the things that actually maybe I didn't get to highlight in this talk, but I'm very excited about, are 2 newer techniques for me that this CDA will bring out. The first being the DTI measure to track structural connectivity. There's really a lack of what we're seeing functionally in the networks to what structurally is happening. And so hoping to bridge that gap, I think could be very relevant to human researchers, where we can get an MRI done, and being more specific in the pathways that are dysfunctional is always helpful to human research. So that's one new approach that I'm really excited to bring with the CDA. And then the second is to use the TMS and brain stimulation approach. And I think if we can complete this circle of finding relevant biomarkers that we think are predicting behavior and actually changing those through a TMS-like approach that could be used in humans, I think that would really bring full circle the translational ability of our recording techniques. I think Susan maybe has her hand up. Go ahead.
Yeah, great talk, Miranda. Thank you. It's nice to see all your data. Following up on your TMS, how are you actually going to do that in the rodents? And like, how specific can it be in terms of a pathway to like probe some of these circuits that you've identified with the physiology?
Yeah, that's a great question. And I think something that is actually going to take much more trial and error experimentation than I've kind of laid out for the purpose of this grant. But in the CDA grant, I proposed to do baseline TMS stimulation on one candidate electrode. For now, that looks like the lateral orbital frontal cortex, where we see this really robust beta response in sham animals that's lacking in the TBI animals, might be a good target. So I propose to start there and do stimulation at beta frequency unpaired to behavior, and then run them on the behavioral assessment, repeat that across 10 consecutive sessions, and then record again to see if that had any changes in their beta frequency activity.
So you use the existing electrodes that are there?
Yeah, exactly. Use the existing electrodes, which does give us flexibility to move to different sites if needed. I don't think any of the sites that are exactly within the region of damage would be very helpful. It's, huge damage that I'm creating, maybe in the mild animals, but certainly not in the severe animals. So we would have the flexibility to move. I think what we should be doing on the back end in our lab is to better characterize these areas in a way that is a causal manipulation. So whether that be optogenetics or DREADS or some sort of causal manipulation, I think that could help inform what the better or best targets are for brain stimulation, and if single-ended stimulation is enough, or if it should be stimulation that's eliciting a specific pathway. So I think those are still some of the questions to work out in order to refine the technique.
Yeah. I had another question, but now I can't remember what it was. So I'll let
I talked too much, sorry.
I think we're just getting towards the end. Susan, I'll just comment that, TMS per se is hard to, people do it directly translate and use like little rodent TMS coils. And the focality of that, it's hard to engineer a magnetic coil small enough to get focality the size of a mouse and even the size of a rat. So that's why Miranda is trying this electrical approach, thinking, well, what we're really trying to model with TMS is the electric field that's generated by it. That's like what we think, at least, is the main thing that we're doing. So it's much easier to deliver focal electric fields. And that's large literature of intracortical microstimulation for that. And, the amount of energy and the distance traveled is very well characterized. So, I mean, in that sense, it's not a perfect model of TMS, but it's a model that we think is relevant.
Yeah, but I mean, you have the ability to be more specific with it. So why go to something that looks more like the human if it's going to be not give you a very good answer
from a circuit level? Yeah,
right, on the circuit level. Yeah. So one question I had was, it seems like a lot of your TBI animals, and you're mainly were showing us lateral OFC, right? So, but just across the different frequencies, it looked like everything was sort of reduced. Is it because you might be near the tissue that's not really there or damaged, or why, or is it just sort of in general, like what do you think about that? Or maybe I'm not
that's a great point.
Person,
no, that's a great point, and I can definitely see how that's the general takeaway from these slides. But I think the first point on that is that I showed, I probably could go back to it easy enough, but it was not just LOFC, but actually many other regions that show this pattern, maybe not at the level OFC does, but still showing that pattern. And some are nucleus accumbens, ventral striatum, very distal from the area of impact. So
Right.
either the whole network is having this power loss function, or it's more widespread than that. And then the second point is that here I'm, clipping out very specific times, first of all, of interest and frequencies of interest. So not all frequencies show this, and high gamma is actually the best indicator where there isn't a huge power loss at high gamma frequencies. High gamma is actually believed to be the most representative, if anything, of single unit activity. So that would argue perhaps against that theory that there's just tissue loss and activity loss.
Right. Yeah.
And then third, we could look at different time windows of interest and show that different frequencies are modulated differently, and it might not always be a power loss scenario.
Yeah, that makes sense. Yeah. It's a shame that so Alex Chu was here, and Vicky and I met with him when he was visiting at the VA, and he was, they have this place where they do these field TBI, and I guess Dakshin, you had talked to him before, but it's only mice at this point. Is that right? Because that would be cool to have a better, TBI that's not just this.
Totally.
Right.
More of
Right now it's only mice.
Yeah.
And it was like complicated to have them. When I talked to him, it was complicated to do it and then have them send the mice. They're like,
Yeah.
send your equipment there and we'll do the stuff.
You're like,
Which is-
it's not so easy.
Not so easy. Sam, you have a question? Or
Yeah,
Abe?
or Abraham. So yeah, I mean, it's not a very well-formed thought, but one of the things that I've been excited about is that we can use genetics to predict phenotypes in animals, right? And maybe you've heard me talk about this, but we've run lots of these genome-wide association studies, and that becomes training data. So now we get new animals, and when they're weaned, we collect DNA, and by the time they're 50 days old, we could predict what their phenotype might be. And so let's say you wanted animals that were predicted to be very high or very low for delay discounting, which it sounded like was one of the things you were interested in.
Yeah.
We could do that. And every 3 months or so, we have 500 or 600 rats. And so we would give you, say, the 5% highest and lowest from those 500 animals. So they'd be relatively extreme. And then I guess, and here's where the thought is really not very well formulated, you could record or do whatever magic you do and see if you were able to observe differences in the way the brain was acting in these animals that have a genetic predisposition to be different. And I wonder if that's interesting and maybe something that you and I would talk about as a follow-up. And it could be delay discounting, but it could be a lot of other things, particularly substance abuse.
Yeah, I love that idea. I think that's kind of, I wish I had time to talk more about this drug study, but that's something we can talk more offline about. And certainly Sam knows some of the details of it too. But we really, have been studying behavior that is not so different between these individuals in order to capture these robust biomarkers. But I think you're right. Now that we have this set, let's put it to the test. Does it differentiate between these clinical populations? We are seeing even some variations between males and females naturally on this test. And so we haven't quite done the analysis to look at that yet, but we're very close. And so that was kind of our first stage of looking at something like that in a population that does naturally have some differences on this task. Does that hold up in the brain signatures that we're looking for as well? But I would love that idea. I think that's a very interesting approach to get really two clear groups. And yeah, what does the brain say about that? And can we manipulate it in maybe predictable ways?
Yeah. I mean, it's nice because oftentimes people will look at like 2 inbred strains or something, and they might be different for whatever you're interested in. But of course, they'll have a ton of other arbitrary differences just because they're 2 inbred strains. And so doing it this way, it's not confounded in the same way. And that's the nice thing. If you see some other difference, like in the brain activity, it really should be genetically related to the thing they were predicted on. So anyway, okay, good. Yeah, well, feel free to reach out, or maybe I'll reach out if I'm organized enough.
Perfect. Great. I can do that.
I think we're over time. So unless there's any last minute questions, I think we can end the talk. Thank you, Miranda.
Thank you, everyone. Thanks for coming.
Her file will be going for review. So anybody who's an associate professor or above, please vote at that meeting. Thanks, everyone. Thanks for coming.
Thank you.